Great. Welcome, everyone. Thank you for joining us today. This is our February edition of the Statistics Former Student Network webinar. We're very happy to have with us Dr. Eileen King. Dr. King is a research professor in the Division of Biostatistics and Epidemiology at Cincinnati Children's Hospital Medical Center. She's involved in multinational and multidisciplinary clinical research studies and provides biostatistical leadership in the design, analysis, interpretation, and reporting of research results. Her areas of research include statistics, data management, and data coordination for the clinical research networks. Prior to joining Cincinnati Children's, she worked in the pharmaceutical industry and was also involved in drug development process. The data management and coordinating centers strengthen the ability of research networks to understand the complex pathways characterizing etiology, diagnosis, and management of diseases and the network's ability to assess clinical trial readiness. Their lab is the Data Coordinating Center for the Rare Disease Clinical Research Network and the Coordinating Center for the Bench to Bassinet program. She's a fellow of the American Statistical Association and has served on the board of directors for the ASA. She's also been a statistician for more than 30 years and began working at Cincinnati Children's in 2009. She received her PhD in 1988 under the advisors of Jeff Hart and Tom Worley, and she's also a recipient of the H.L. Hartley Award in 2011. Thank you, Dr. King, for agreeing to share with us your statistical background and knowledge and help grow our students. I appreciate your taking this time with us today, and please take the floor at this time.
Great. Thank you, Marcy. Everybody can hear me okay?
Yes.
Okay, great. So welcome to all my Aggie colleagues. I'm excited to be talking to you today. I'm going to talk to you about my path about changing the world with statistics. In the beginning, I graduated from St. Patrick's High School in the Panhandle, Nebraska, Sidney, Nebraska, 1972. And in the 70s, everyone was out to change the world. So as everyone was, I was ready to change the world. I had no idea at that time, actually, that a career in statistics even existed. So I went to college in Denver at Regis College and got my Bachelor of Science in Psychology, but also loved mathematics, so double majored in psychology and mathematics. And after that, I taught high school. I taught 7th to 12th grade math in a small school district in northeastern Colorado, and decided that teaching 7th grade and 8th grade math was probably not my calling. I wanted to teach. I didn't want to discipline. So this is where I say my journey with statistics began. I was introduced to a gentleman who eventually became my brother-in-law, married my sister. He had gone to the University of Wyoming and had gotten a degree in statistics. And I thought that sounds very interesting. And so I applied to the University of Wyoming, got my master's degree in statistics, also met my husband Dennis there, who also is an alum and got his PhD from Texas A&M University. And then began my I say that's where my statistical journey began, because that's where I started to teach statistics while I was in graduate school, got my master's degree. And at that point, we moved to the University of Arkansas. And at the University of Arkansas, we were employed at the University of Arkansas, and I taught math. And I taught statistics courses in the math department and worked in the math stat consulting lab. And we were there for about 5 years. And at that point, I determined that there were a lot of things that I did not know. And I needed to advance my training, my knowledge in statistics, statistical theory. At the University of Arkansas, I crossed paths with two people who actually were very instrumental in my career. One was Dr. Jeff Hart. He was there teaching. He had gotten his PhD, and he was there teaching at the University of Arkansas. While we were there, he actually moved to Texas A&M University. The second individual I crossed paths with while I was at the University of Arkansas was a man named Michael Meredith. And he our paths crossed when he passed his teaching materials to me on teaching statistical programming. And it came to be that by crossing paths with him, that led to a career at Procter & Gamble. And I'll get to that in a little bit. Then went to Texas A&M University. Obviously got my PhD there under, as was said, Dr. Hart and Dr. Worley. During my whole career, since I've gotten my master's degree, I also want to point out that I've also been very connected with the American Statistical Association and was very lucky to be able to really participate in a lot of the committees, et cetera, in ASA, including being elected to be on the board of directors. After getting my degree at Texas A&M, I got a job offer at Procter & Gamble in the Procter & Gamble company. Now, Procter & Gamble is more than a pharmaceutical company. It obviously is a consumer products company. And this is one of the reasons why I chose Procter & Gamble as the company that I wanted to go to work for. And that is because it was more than just pharmaceutical research. And I'm just going to talk a little bit about a few of the projects or areas that I worked, whoops, I went too fast, that I worked in. And then I want to talk a little bit in depth on the pharmaceuticals area. So like I said, Procter & Gamble is a wonderful company to work with. I totally enjoyed my career there, but they had many different research areas that you could be involved in. And I was always involved in the R&D. I did not get involved in marketing or advertising or any of those areas. One of the areas I got involved in obviously was healthcare. And this is over-the-counter healthcare and also pharmaceutical healthcare. And one of the projects that we worked on was trying to develop a new nasal decongestant that would be more effective than pseudoephedrine, which is the product that was on the market at that point in time. And the one thing I want to point out there is that, you know, you learn as you work in these areas different things. And one thing I did learn was that the ferret has a similar nasal system as humans. And so we did our preclinical work in ferrets where we would infect them with a virus. They would become congested. We would use technology to measure their nasal airway resistance. And that is how we would test our compounds that we were trying to determine would they be effective to be a new nasal decongestant. That's how we would test our compounds in the animal model. Other areas that I got involved in healthcare on the over-the-counter side was especially involved in all of the Vicks cough and cold products. The other piece that I was involved in while I was at Procter & Gamble, if we go down here to where it says family care bath tissue, is that we were in the research and development area, but also claims and commercials are made based on, you know, data that we generate from the studies that we design and analyze. And I just wanted to point out that while I was there, this is when the claim or the commercial about fewer pieces left behind with Charmin came about. And I just want to point out that when companies make these kind of claims, they have to have clinical research studies to back those claims. And if they don't, they will get, the other companies will try to make them take those claims off. And as I was preparing for this talk, I actually found out that one of the competitors to Procter & Gamble was actually challenging this particular claim over time, which leads me to another area that I got involved in at Procter & Gamble, and that was providing statistical support, statistical guidance and input when claims were challenged. We would have to read depositions. We would have to look at the data of our competitor. We'd have to look at our own data. If we were being challenged, we'd have to make sure that our data really supported our claims. If we were the ones doing the challenging, we would have to try to find holes in the data that they made available to support their particular claims. One of the other areas Procter & Gamble was involved in or had while I was there was in the area of pet care. They had acquired IAMS. And it was very interesting working in this particular area. And just as an example, if you want to make a dog food that helps to make your dog more vigorous, moves, more active, etc., you have to have data to support that. And so we did studies where we would put activity monitors on pets. We would design research studies to determine if a dog was given dog food by a competitor or an old version of our own dog food versus our new formulated dog food, which is really supposed to make your dog a lot more active and vital. We would have to design those studies, analyze those studies. And so I looked at activity monitoring data for dogs and did analysis on that. It was very interesting because as you looked at those data, you could see when the owner of the pet came home, because you really did see obviously the change in activity when at the end of the day when the owner came home. That's just a flavor for some of the different kinds of I'd say consumer products that I worked on, or the different kinds of work that you do in a consumer product research company. What I want to get into a little bit more in depth is the pharmaceuticals, because I became involved in pharmaceuticals at Procter & Gamble. And this is an area that I especially loved because I was able to work with a very diverse team of individuals. We were in R&D, we were very collegial, and it was very much a multidisciplinary collaboration to get pharmaceutical products approved. Worked with very brilliant medical doctors, clinical research associates, data managers, medical writers who would help us with writing of the protocol, with writing of manuscripts, etc. And it was a very fun and exciting time to be, or exciting career opportunity for me. But I want to go through pharmaceuticals and the drug development process and the role that statistics plays throughout the entire process. And this drug development process schematic you can find on the FDA.gov website. And if you Google the drug development process, you'll come to this page. This is just a screenshot. And it shows you the steps that are involved in getting a drug approved. And I personally, in my career, have been involved in every single one of these steps. Maybe not for the same compound, but for various compounds that happen to be in different stages. So the first step is the discovery concept stage. So you're going to research for a new drug, and this also talks about devices. I did not work on devices at Procter & Gamble. I've worked a little bit on them at Cincinnati Children's. But you do the laboratory research where you're trying to find the compound that you think will have an effect on whatever it is you're targeting. Once you have identified that compound, and that also involves being involved with statisticians who will help you to analyze the data that you're generating when you are developing those compounds. Once you identify a promising compound, it goes into preclinical research. And this is where these drugs or devices undergo laboratory and animal testing to answer basic questions about safety and also about efficacy. So I was involved in trying to come up with a new pharmaceutical product, like I said, for a new decongestant. And so we used the ferret model to help us to determine did this particular compound or a tweak of that compound show us efficacy. And then you do animal studies for safety. So you have to do all your toxicology studies. Those are pretty well, how do I want to say, from a statistical perspective, the toxicology studies are very well defined, kind of rote. we didn't really get too involved in those because they had all the statistics already down, and they just kind of reran them all the time. But depending upon what disease area you were trying to target, the animal studies for efficacy may change. And I got very much involved in animal studies. Like I said, one example is the ferret with the nasal airway resistance. Then you move to testing that drug or device on people to make sure they're safe and effective. And I'm going to get into this a little bit more detail. Then you have to gather all of that data and send it to the FDA for their review. And then once the drug is approved, the final step is the post-marketing safety monitoring step to make sure that you didn't miss something while you were developing your drug once it got onto the market. So this also is from the FDA website, and this goes into a little bit more detail of what I just showed you. So once again, the drug is developed, the animals are tested. You have to put in an IND application that provides data that says, go ahead, you can use this compound in people. It's safe enough to be tested in people. And then you go into clinical research testing. And I want to point out a couple of things just from this slide. The clinical research testing generally has about 3 phases: phase 1, phase 2, and then phase 3. Sometimes you call these first 2 phases the learning phase and the last phase the confirming phase. But in this schematic, I want you to notice something, that the numbers in phase 1, where you're trying to determine, okay, is this compound going to be safe? What's the highest dose we can use for safety reasons? Here it says 20 to 80. Then you move to individuals in your studies. Here you go into the phase 2, and it says a typical number of patients used in phase 2 studies is about in the hundreds. And here you're trying to determine, will this drug work? Is it efficacious in the disease that I am interested in? Will this new decongestant actually work with people who have a cold or allergies? And then once you feel pretty comfortable that the drug is safe and effective, then you have to do your confirming phase. And these are your phase 3 studies. This says thousands of people. And this is where you run your well-controlled studies. You know what the dose or potential doses of the drug are. And you really are confirming that this compound is safe and it is effective in patients. The reason I'm pointing out these numbers is, as was said in my bio, I'm now in rare disease research. And there aren't, in some of the diseases we have, there aren't even 100 people who have this disease. So how do you get new drugs approved in a disease, an ultra rare disease, where I'm told by the investigators, we've only got 50 people worldwide, but we still need a treatment. I'll get to that a little bit later. This schematic just talks about the review process. We submit an application to the FDA and it's reviewed, and then the post-marketing. But I want to step back a little bit. So I want to talk about this and again, highlight how much statistics is involved in all of this. This is a drug, and I found this again on the FDA website, if you know where to look. This has to do with the last project I was involved in at Procter & Gamble, where we were trying to get an 800 milligram dose of mesalamine, which is used for the treatment of ulcerative colitis, and we wanted to get that approved. We wanted to, I'll talk about a little bit more about that. But this is the label that we negotiated or that we worked with or provided data for the FDA that they gave us at approval. So once again, you start your drug development process by, if you're doing a drug, they come up with a new molecule, a new drug. And then you say, okay, this drug is going to work. And in this case, we want to say this drug is going to be effective in patients with ulcerative colitis. So just very, very briefly, this was kind of what this case study is all about, because this is what the label is all about. We wanted to determine the safety and efficacy of a 4.8 gram per day dose of mesalamine, or our brand name was Asacol, using 800 milligram tablets in patients with ulcerative colitis. Our dosing was going to be, we're proposing was 2 800 milligram tablets taken 3 times a day. The situation we were in is that we already had an approved dose of 2.4 grams per day, but this was using a 400 milligram tablet, which meant patients had to take, if we were going to go with the 4.8 gram, they were going to have to take a whole lot of these 400 milligram tablets. But this was already approved. And so if we wanted to get this new dose and new dosage form approved, it was not going to be ethical nor practical to include a placebo group in our study design. So keep that in mind a little bit. So again, here is just a little bit of the label, and I'm going to get to the table of contents of the label in just a minute, but when they do the label, that you don't have a trade name at the time. So they say trade name is indicated for the treatment of moderately active ulcerative colitis. So this is after we got approval. Safety and efficacy of Asacol HD beyond 6 weeks has not been established. And the reason I'm pointing some of this out is because your study design that you do is going to dictate the verbiage that is in the label for this particular drug. And the label is very important, obviously, for your medical providers if they're going to prescribe your drug. Okay. And again, use beyond 6 weeks has not been evaluated. Our primary endpoint was at 6 weeks. And I think that's it for right. Oh, and the tablets could be taken daily with or without food. So for every little piece in this label, we have to have data that supports all of these statements. So when you are going to get a drug approved, you need to think about, we always think about what is the label going to look like? And these are the components that you have to have in a label, and you have to have data that the FDA can use in order to fill in every part of this label. And statistics plays a key component in designing the studies, analyzing the data, and making sure that the data are summarized appropriately so that you can fill in every piece of this label, from adverse drug reactions. Are there any drug interactions? How do you use it in specific populations like pregnancy, pediatric use, geriatric use, clinical pharmacology? What are the pharmacokinetics and mechanism of action? What's the safety, the toxicology of this? And then, of course, the clinical studies. So just a little bit on this. For those of you who don't know pharmacokinetics, pharmacokinetics tells you how the drug is absorbed, distributed, metabolized, and excreted. And I've been very much involved in pharmacokinetic studies. So you give individuals the drug. In this particular case, it's a pill, so the drug is absorbed, and then it is excreted eventually. And we need to understand what the pattern of this is, because this dictates how you will dose the drug. How frequently will you dose the drug? If it's excreted very quickly, then you have to dose it more frequently in a day. If it is excreted and your half-life, which is how long does it take before half of the drug is out of the system, if that half-life is very long, then you might only have once a day dosing for the particular drug. So this particular schematic just shows you for 2 different products what the pharmacokinetic profile is. From a statistical point of view, we calculate parameters that are very much interest to the scientists. Area under the curve of this pharmacokinetic curve, we have to calculate that, and we compare it between 2 different maybe formulations or 2 different products. We also calculate the maximum, so that we know how quickly was it absorbed and what's the maximum amount that gets into the body. And then we calculate the half-life, which is based on, if you do this curve as a log transformation, and you look at the slope of the curve when it's being excreted, that gives you information on the half-life, which dictates the dosing. I'm going to go through a bunch of slides pretty quickly here, but I just am trying to point out that how much statistics comes into play with this. And even though the label, some of these labels are fairly long, but this is like 10 years of work to provide all of the data that have to go into the label. So this particular part of this label for azokal is the pharmacokinetics section. And I just wanted to point out, you know, we have to provide the data for what is the area under the curve and the Cmax and the half-life for both the mesalamine, which is the drug itself, and any metabolite that might be, that your body might make out of this particular drug. And that has to be included in the label. Actually, nowadays, I see this as mean and standard deviation. Probably nowadays, we mainly would put in here geometric mean and 95% confidence interval because the data are very skewed. And the geometric mean will be estimating the median level of mesalamine for AUC, the median level of the AUC for the mesalamine dose. The one thing I want to point out is down here, the statement that where my pointer is, where it says the 800 milligram tablet has not been shown to be bioequivalent to 2 azokal 400 milligram tablets. We tried to show bioequivalence and we could not. And the reason we could not is because azokal has a special coating on the tablet so that it will not dissolve until the pH in your system is at, I believe it's 7, which means it has to get all the way down to the gut. It will not metabolize in your stomach. It has to go all the way down to your gut to metabolize. What happens when you do that is your area under the curves and your Cmaxes for individual patients are so variable that you cannot show bioequivalence. And bioequivalence means I have to prove that the area under the curve, if I give 1 800 milligram tablet versus 2 400 milligram tablets, that the areas under the curves for both of those, if you go back up here to this, that these two curves would look exactly, very close to exactly the same. We had too much variability in our data, so we could not do that. That would have been the fastest way to get this approved, but we just could not do that. And it says because the mechanism of action of mesalamine appears to be topical, that's part of the reason why it leads to problems with bioequivalence. This particular information actually came from the medical and statistical review of our submission. So we submit an application, we provide all of the efficacy and safety data that we have available for this dosage form and this drug, and it goes under a medical review and a statistical review. And your statistical reviewers at the FDA are very important and they carry a lot of weight, and what they say really does impact decisions that are made. And we have had applications denied because the statistical reviewer did not agree with the data that we had provided. And rightly so, they're doing their job. They have a job to do to ensure that whatever a drug is put on the market, it is safe and is effective, and that the risk-benefit ratio is very well understood. So we did a study. We ended up doing an efficacy study where we actually compared the 4.8 gram per day dose to the 2.4 gram per day dose. It was a blinded study. The 2.4 gram used the 400 milligram tablets, the 4.8 gram used 800 milligram tablets, and we compared those two. What I want to point out, though, was when we did the study, you always have inclusion criteria. And it's important to realize that the inclusion criteria in your clinical research program dictate who can use the drug. So you'll notice that we only enroll patients between 18 and 75 years of age. So no, we can't use this in the pediatric population. We don't have any data in that population. We can't make inferences to that population. The other big thing is these individuals had to have a confirmed diagnosis of moderately active ulcerative colitis. We didn't enroll any mild ulcerative colitis patients. So it's not, your label will not be indicated for mild because we don't have any patients in that with people with mild ulcerative colitis. So if you pay attention to your commercials on TV, you'll notice a lot of times they'll say this drug or biologic is approved for, is to be used in patients who have moderate disease. And that is because that's all they enrolled in their phase 3 clinical studies. So that's a very important thing to realize when you're designing these studies. We ran the study. We compared the success rates of using 2.4 gram per day and 4.8 gram per day azocol. This is a long story, and I don't want to spend all my time on P&G. We actually ended up doing what's called a non-inferiority comparison, and the reason is as follows. We could not use a placebo in our study. All we wanted to do was show 4.8 gram per day was efficacious. We could not put a placebo in there to show, and that's how you would typically show 4.8 gram was efficacious. But people with ulcerative colitis should not be given placebo because there are approved products on the market. So we negotiated with the FDA to say the following. 2.4 gram per day we know has been proven to be effective. So it is superior to placebo. If we can show that 4.8 gram per day is no worse than 2.4 gram per day, we can claim that 4.8 gram is also effective and would have beat placebo, would have been superior to placebo if we could have included placebo in the study. The FDA agreed with that and said, yes, you can use that approach to say that 4.8 gram is an efficacious dose. Can't say it's better than 2.4, but we could say it's an efficacious dose. We actually designed the study with a step down with a closed testing procedure where we said, well, the first thing we're going to do is we're going to determine non-inferiority. So as long as 4.8 is no worse than 2.4 within a certain margin, we're going to conclude that 4.8 is efficacious. If in fact 4.8 beat 2.4, then we could have the additional claim that the higher dose is actually better than the lower dose. Well, you can see with the p-value, 4.8 did not beat the 2.4 gram per day, but when you develop the 95% confidence interval for the difference between the two, we said that our safety margin, our lower margin was going to be 10%. So as long as we were, this is percent in remission at 6 weeks, as long as we were no worse than, that the lower limit was no worse than 10%, we were going to claim that we were non-inferior. The FDA had a little bit of argument with that margin, but the fact that our margin was only 1.9% worse in the confidence interval than 2.4, they said clinically those two doses are the same, and that allowed us to get approval for the efficacy of 4.8 gram per day. Now you might argue, why would you take that over the 2.4 gram per day? That's a different lecture. I just wanted to point out that all of this data goes into your label. You see down here, you've got the primary endpoint with the data that I just showed in that particular table. You can see, I've already showed you that before. I got to move on. The one thing they were very concerned with, because we had already done 2 studies and we didn't have sufficient evidence to prove the efficacy of 4.8 gram per day. In our 2 previous studies, there was an indication that males and females were reacting, had different outcomes that they didn't seem to be consistent. So we specifically, prospectively said we were going to look at the outcomes in males and females and really look at the consistency in those two groups. And so in every application, you always do these kind of plots, which shows how consistent your results are across various subgroups. I know the quality of this isn't too great, but you have males versus females. You have less than 65 versus greater than or equal to 65. See, confidence interval for greater than equal to 65 is very large. That's because there were very few patients in our study in that group, and so on. This is just an indication of when we actually looked at success rate in males and females in this particular study, they were quite comparable. We also have to provide data about the adverse events on, for a particular drug. So we did it for 2.4 and 4.8, and there really wasn't any difference. So 4.8 gram was no more of a safety concern than 2.4 gram per day. We provide all of this data. Your statisticians summarize the data, provide all that data for the label. And this is what, when you listen to all of those side effects that they talk about on your commercials and they do that laundry list, it has to do with the data in this particular table. We didn't study pediatric patients. We didn't include a sufficient number of subjects aged 65 and over to determine if they respond differently than younger subjects. That's in the label. And I thought I just wanted to put this up because life is not a straight line from point A to point B sometimes. We had done 2 studies before. The statistical reviewer did not agree or stated that we did not provide substantial evidence demonstrating superiority of 4.8 gram over the 2.4 milligram per day dose. Actually, that's gram per day dose, I believe. In the previous studies. And that's when we changed our design in this new study to be a non-inferiority study. So when we resubmitted and added the extra study, because when we submitted the first time, the response we got back was you need to do another study to provide, to confirm what you've already are claiming in your first 2 studies. Then he did say, thus Azicol 800 dosed at 4.8 grams per day should be considered efficacious compared to placebo. So we finally got it. I got a nice backpack on this project. I used that forever, and it was a very fun project. But that's just one example of what is always involved in getting a drug approved and how much statistics plays a role in that whole process. So even though that label is not that long, you're talking about 10 years of work and lots of data and lots of input by your statisticians on that. At P&G, I was also involved in managing and managing an organization. I was the head of the Department of Biometrics and Statistical Sciences toward the end of my time there. And I just wanted to point out a few things I learned at P&G. P&G is great because they do a lot of training for their managers. So when you're managing and leading, a lot of it has to do with coaching. You have to coach the people that you're leading, you're managing. You have to continually teach them not only about statistical principles, but about interacting with people, how to be effective, how to be influential. You have to be concerned with people's career development because everyone wants to develop in their career. They want to move forward. They want to see that they're making progress. They want to be involved in more and more challenging projects over time. And you have to provide project oversight. As a manager at P&G, I was also highly involved in strategy setting for the whole development program from from preclinical all the way down to post-marketing. Obviously involved in working with all of the managers of all of the other functions on progressing projects. How do you effectively bust any barriers that people have? And my opinion, if you're managing an organization, a technical organization, you need to have technical oversight. So people with the technical knowledge should be managing. People should be managed by people who have the same who have the technical knowledge that they're managing up to a certain level. Leadership skills, technical knowledge, you've got to have good oral and written communication skills. You have to be an effective delegator. You have to know and provide good guidance on what you're delegating. What are the boundaries that you have, that these individuals have to live in? If you give them a project to do, what are the boundaries? What are the showstoppers so they don't go down a path and then you say, oh, no, you can't go down that way. Leveraging diversity. And I had this on here before the diversity movement, which has taken place in the last several years, but it's really important to you leverage diversity. And I don't mean just diversity you can see. I mean diversity of different cultures, diversity of ways of thinking, etc. How do you leverage that diversity? And you have to listen to people and learn from them. Both the people that you work with, both the people that you manage, etc. And then knowing when to compromise, because sometimes you do have to compromise. I know statisticians sometimes don't like to compromise, but sometimes you have to say, you know, a t-test is going to be good enough for this, or yeah, you could do it that way. Okay, that's my career at Procter & Gamble. So I retired from there after almost exactly 20 years to the day. And then I did some interim teaching at Miami University, their Hamilton campus, for about 9 months. And then I was lucky enough to get on at Cincinnati Children's Hospital Medical Center. Went to a wedding with someone at P&G. One of my colleagues' wives works at Cincinnati Children's, said, hey, we're looking for a faculty statistician. And the rest is history. Why did they bring me in? I wasn't a typical person that should go to an academic research center. I didn't have a publication record. You don't necessarily get a strong publication record in industry, especially if you're in the management track. But what they found out was that I did have this background in the pharmaceutical area, and I did know how to lead, and I did know how to manage, and I did know how to build organizations. So when I interviewed, they said they want to build capacity to successfully compete for data coordinating center grants. And I said, oh, so I asked the thing, you want to do that? And he goes, well, why shouldn't we be able to do that? I'm like, okay, I didn't say this, but you have no infrastructure, but that's okay. And then also collaborate with CCHMC researchers, because as you know, in academic healthcare research and all research, there's not enough statisticians to go around. And so I did collaborate with a lot of CCHMC research. But I want to talk about DCC opportunities, because I want to let you know what the NIH is. So now I'm working with the NIH. Worked with the FDA before, now I'm working with the NIH. The NIH in the recent years has determined that they want to pull together these disease-specific networks, and these disease-specific networks should have a data management and coordinating center. People who know how to manage data, make sure you're having high-quality data, that it's clean, well-organized, findable, etc. Also, the NIH determined that they're giving grants to investigators to do clinical research studies. And most of these investigators don't know a lot about data management. They don't know, the investigators themselves know some statistics, but they're not experts in statistics. And so now if you ask for over a certain amount of money, and I believe it's $500,000 a year, you have to put in a DCC companion grant. And this has to be led by someone who knows data coordination, data management, etc. There's also these multi-site learning networks and registries, and I'm going to talk a lot about those coming up. And then multi-site prospective clinical trials sponsored by industry, in which the PI is affiliated with CCHMC and UC, and I'm going to give you a quick example of that. So the first thing we did was we got all together. I did not do this by myself by any stretch of the imagination. I had some great colleagues. We had to hire the people who are going to staff our data coordinating center. We had to make sure that there was a career and professional growth opportunities for these individuals. we had to write SOPs, figure out what the processes were going to be. Luckily, I'd been at Procter & Gamble. I was able to get some SOPs from my P&G colleagues that we used as templates for that, and come up with the technology, especially data capture technology, to collect our data. You don't collect clinical research data in Excel spreadsheets. They need to be in some kind of a system where you know who's changed the data. People can't inadvertently change data when they don't know it. You know, if you copy lines or of an Excel spreadsheet from one to the next, etc. So we had to build that infrastructure. One thing I learned at Cincinnati Children's was if you're doing pediatric research, it requires multi-institutional collaboration. These are just some of the areas I've been involved in, but all of my colleagues here at Cincinnati Children's, they work with, for the most part, with institutions beyond just CCHMC. I'm going to go through some of these a bit. So I want to talk a little bit about some of the projects I've been involved in. Improved Care Now is in the area of Crohn's disease, ulcerative colitis, a great fit from what I did at Procter & Gamble. It is a collaborative community where clinicians, researchers, parents, and patients all work together to come up with care for healthier children and youth. And so Improved Care Now, I'll just point out a couple of things. It has 970 pediatric gastroenterologists that have joined this collaborative community. They meet, I think, twice a year to all come together at not all these 970, but where patients, researchers, parents all come together and they talk about the disease and how they're going to make it better. These are some of the targets that they're trying to get is what percentage of patients are in remission, what percentage of patients have satisfactory growth, etc. There's a whole website on this. It's called the Improved Care Now. Interestingly enough, they use statistical process control methods in this particular collaborative. They use control charts. So what they did was they tried to standardize the way that you care for these patients, and then they were tracking outcomes over time. And this particular outcome is percent of patients in remission. They had a target at this, when I ran this graph of about, I guess about 83%, and they were trying to move toward that target. So they only started at about 67%, and then they moved, they were moving obviously up close to the target. And I think they've actually over time have this first thing I started on it at Cincinnati Children's, they've actually improved the target. So you wouldn't think statistical process control methods would be used in academic medical center research, but it certainly is. It's used a lot. The other one I'm involved in is the Pediatric Obesity Weight Evaluation Registry. I was involved in Improved Care Now. I saw how that worked. One of my colleagues said, hey, I want to start a registry in pediatric obesity research. And so we want to improve the health outcomes of adolescents with overweight and obesity. It's a disease. These patients have overweight and obesity. They are not overweight and obese adolescents. They are adolescents who have a particular disease, and we're trying to figure out how to make their life better. These particular programs address nutrition, physical activity, and behavioral strategies. However, over time, new drugs are getting approved to treat this disease. And I want to point out that I collaborate with people all over the country. The states in purple are the states that actively are participating in our registry. And I believe the cycle was probably 20 to 22. We started this in 2014. Every 2 years, these institutions will pay money in order to participate in this registry. They pay a small amount of money to pay for the data coordinating center support for this. Every 2 years, we go out, we say, do you have the funds to continue? We recruit new sites. We reevaluate what data we're collecting. We modify our data collection forms, and we have just started I believe our 5th cycle of this but you can see that some sites come and go in this particular registry. We are tracking outcomes, and this happens to be a weight metric outcome, and we provide this to the sites so that they can see a site data are in red, and the rest of all the sites combined are in blue. And so they can compare how are they doing at 6 months, 4 to 8 months in this weight outcome metric. Negative means that they have actually improvement in their health status. They've actually improved their BMI, negative. I also worked with a colleague at University College of Medicine, and we worked with this says it all in the blue. We delivered a dietary intervention at the supermarket. So many of the Kroger supermarkets have what's called a little clinic in there. And so we used the services of the little clinic to help track some of the health outcomes of participants in this study, but they also were able to work with the patients on what food should you buy, etc. So I'm going to show you that in just a second. Dietary intervention delivered in the supermarket offers convenience, accessibility, full food inventories, and increasingly in-store registered dietitians, online shopping, and delivery services. We designed the study. We had 3 arms. One was just get medical nutrition therapy. Second arm was purchasing data guided in the aisles. Someone walked around with them, helped them figure out, here's the foods that you should buy to improve not only your weight, but your cardiovascular status. And then also, if people had access to online shopping, would that help them as well? Because now if you're online shopping, you may not be as tempted to buy the chips at the end of the aisle. This was just some news that we got on this, fixing America's eating habits with effective stakeholder collaborations, and effectiveness of supermarket and randomized control trials, etc. This was the very first large data management coordinating center grant we got. I believe it was like $25 million or maybe $32 million over 5 years. I'm running out of time, so I'll just say this is in the area of congenital heart disease. And we supported the Cardiovascular Development Consortia, which was doing animal models, zebrafish models, etc. And then also the Pediatric Cardiac Genomics Consortia. And they were trying to understand the genomics of congenital heart disease. One of the studies that they're doing is enrolling participants with congenital heart disease and their parents, and they're trying to figure out what are the genes that are affecting, that are related to congenital heart disease. We provided administrative coordination and also data hub services. And the main thing in this little box here is this data is available for researchers to go into this system called HeartSmart. They can download the data, they can use it to do analysis. And then we also put the data out for the general research community to use. So this is all genetic data here, with also some phenotype data with it. We are now, this was the first 5 years, we just got renewed on this. Now what we're trying to do is correlate the genetic outcomes with clinical outcomes. So we are extracting data from electronic medical records and using the appropriate regulatory processes, et cetera, very, very much in tune with that. And then we want to try to determine what is the relationship with some of the genetic findings we have and what their health outcomes are. And the last thing I'm going to talk about is the latest data management coordinating center grant we got, and that's the Rare Diseases Clinical Research Network. What are rare diseases? They affect their diseases. They're defined as diseases that affect fewer than 200,000 Americans, but there's more than 7,000 known rare diseases. So about 350 million people suffer from rare diseases worldwide. It's funded by the NIH. And the thing that's interesting here is this network was established by Congress under the Rare Diseases Act of 2002. It's made up of 20 consortia. Each of these consortia study at least 3 rare diseases. NIH is an active partner. Researchers and clinicians are active. And again, here you're seeing patients and patient advocacy groups very active in this whole research process, and is funded by multiple institutes at the NIH. And our goal is to achieve faster diagnoses and better treatments for patients with rare diseases. I've already talked about this, except I do want to say another goal of the consortia, each individual's consortia, is to make sure that they're developing the next generation of rare disease researchers. And then they also support innovative pilot projects. We provide the data management coordinating structures. We have an administrative core, a data management core. This one is focused on how do we make all this data that we're generating in this network available to researchers that aren't part of this network across the country and across the world. We also make sure best practices and protocol development and data management are done. So we review all the protocols, provide guidance and input, and we build their data management platforms for them, or databases for them. And we have a whole core that is supporting patient engagement. And then also research dissemination, getting the patients engaged and disseminating what we're learning. Worldwide and international. This tells you a little bit about what all is involved in this. We've got 273 active sites, 128 active studies. So it's a huge, huge network. We have a nice website. You can Google RDCRN. And we do have a challenge. And as I told you before, way back, it's hard to design studies when you don't have very many patients, and especially to get that magic 0.05 p-value. We are investigating maybe the use of master protocols, which are basket, umbrella, or platform trial designs. So if anyone on this call has experience in this, especially in the area of rare diseases, I think these are used in cancer research a lot, but we need to figure out how we can apply them to rare disease research. Oh, I left that. That slide shouldn't have been in there. And then also how to use external controls. So all of our consortia have a natural history study, which could provide a source for external controls. How do we bring that into the design of our rare disease research? So I set out to change the world in 1972 when I graduated from high school. And when I came to Cincinnati Children's and I realized our tagline was changing the outcome together, I thought, well, I pretty much have met that goal. And so a wonderful career, and I certainly enjoy it. I'd hope to leave a lot more time for questions, but I will entertain any questions, and you can always email me. If you Google my name, you'll find me on the Cincinnati Children's website.
Thank you, Dr. King. It was an awesome presentation. You clearly have impacted the world with your work in statistics. As Dr. King suggested, she's available here now to answer a few questions if anybody has any questions at this time. Please chime in.
Hi, Eileen. This is Mike Longnecker. I was wondering, in both medical schools and in P&G, are there opportunities for students with just a bachelor's degree in statistics?
Thank you for asking me that question. I meant to say, when we built our organization, we hired at all levels, both at Procter & Gamble and at Cincinnati Children's. So we hired PhDs, and we hired at Procter & Gamble and at Children's, and we hired masters, and they work very closely with the faculty at Cincinnati Children's, but they also work, as they develop and grow, they work independently with the researchers at Cincinnati Children's, and then they always have their faculty if they need to go and get some additional, maybe stronger theoretical guidance. Bachelor's level, very much so. We hired them at Procter & Gamble. They did a lot of SAS programming for us, but even in that role, they really, work with the multi-functional team And many of our bachelor's level individuals at Procter & Gamble actually took night courses. And then I eventually worked with Wright State that they could do some remote courses, and they got their master's degree. Many of our master's prepared statisticians would have liked to have had that opportunity to get a PhD remotely, because they had a great job. They didn't want to give it up. So yes, at both Cincinnati Children's and at P&G. And I did want to say is we're hiring at a faculty level, and we're also always an opportunity to hire at both the bachelor's and the master's level. So if anyone's interested, please contact me, especially at the faculty level, because I happen to be leading the search committee.
That's excellent news. Other questions?
And if you're master's or bachelor's, you can also contact me, because I work closely with our managers of our data management analysis center, and I always pass along resumes to them.
Is there room for data science educated individuals, or is it specifically statistics?
No, that it doesn't need. No is no. It doesn't need to be specifically statistics, and absolutely, there's room for data science. So, especially, I've been out of P&G now for 13 years, but I know there's room there as well. Cincinnati Children's, absolutely, because you've got the genomics data. We have large data sets. We have imaging data. We have all this electronic medical health record data that we are using, and data science is also something that's very big. And in fact, Cincinnati Children's actually has a new initiative related to data science. So if you have that, and honestly, if I was starting out, I would probably go into data science. I think it's
I'm
a
at
fascinating
Regis now,
field.
completing my master's in data science, so I might just send you an email later on.
Absolutely. It's Eileen.king@CCHMC, Cincinnati Children's Hospital Medical Center.org. Yep. And if anybody ever just wants another chat about your career, about opportunities, yeah, email me. We can set up a time. I love to talk about careers.
It's a little after 2. Maybe Dr. King, could you entertain one more question if there is one?
I was just
Sure.
want to make a comment. This is Bill Smith. I was department head when Eileen was there, and we're just proud of her as we could be. She's done a little bit of everything in terms of the things she's talked about, but I'll testify to the American Statistical Association. She was actively involved both in chapters and in the board of directors. So we appreciate her and are proud of her to be one of our graduates. Thank you, Eileen.
Thank you.
And your regards, by the way.
You bet. And thanks for our time at Texas A&M couldn't have been more wonderful. And as Dr. Smith knows, we went there, we had a 3-year-old and a 3-month-old when we started at A&M. So we had a lot of support there.
That's just awesome.
Just
And,
one other thing, Eileen, it's 82 degrees. I was just off the golf course, so I'm looking forward to seeing you and Dennis down here again.
we are here.
Oh,
We're in
San
San
Antonio?
Antonio. We've been here since February. He's going to get in touch with y'all to golf.
Good.
And he is actually not listening to my talk. He is on the golf course. He has been most every day this week.
I went out this morning so I could listen to your talk.
So we definitely have to get together to golf. That's
Good.
for sure.
Sounds good. Excellent talk, Eileen.
Thank you.
Indeed, excellent. I appreciate you taking time out of your very busy day to share with everybody in the community all your successes with statistics.
Thank you. Thank you for this opportunity. It's been fun. Good to see everybody again.
All right. Farewell, everyone.
Bye, all.